How do high-risk HPV oncoproteins cause cervical cancer? Imagine a tiny virus that can change the course of your health by sneaking into your cells and disrupting their normal functions. High-risk human papillomaviruses, especially types like HPV-16 and HPV-18, are known to be major causes of cervical cancer. They do this through special proteins called oncoproteins, mainly E6 and E7. These proteins interfere with the cell's natural defenses against cancer, leading to uncontrolled growth and, over time, cancer development. The E6 oncoprotein plays a critical role by targeting a key tumor suppressor protein called p53. Normally, p53 acts as the cell's quality control, stopping cells with damaged DNA from dividing or triggering cell death. When E6 binds to p53, it marks it for destruction. This prevents the cell from repairing DNA damage or undergoing apoptosis, which is a natural way for the body to eliminate potentially cancerous cells. Without p53 functioning properly, damaged cells can survive, accumulate mutations, and eventually turn into cancer. E7, on the other hand, targets another important tumor suppressor called the retinoblastoma protein, or PRB. Under normal circumstances, PRB controls cell division by regulating E2°F transcription factors. These factors are necessary for DNA replication and cell division. When E7 binds to PRB, it disables its ability to control E2°F. This releases the brakes on the cell cycle, causing cells to divide uncontrollably. This unchecked cell proliferation is a hallmark of cancer development. In many cases, the viral DNA from high-risk HPV types integrates into the host cell's genome. This integration often disrupts viral regulatory genes like E2, which normally keep E6 and E7 in check. As a result, the levels of E6 and E7 increase significantly, amplifying their effects on the cell's tumor suppressors. The overexpression of these oncoproteins correlates with the severity of cervical lesions and the progression toward cancer. Beyond disrupting cell cycle controls, E6 and E7 also help the virus evade the immune system. They can modulate immune checkpoints, such as PD-1 and PD-L1, creating an environment where infected cells are less likely to be attacked by immune cells. This immune evasion allows the infected cells to persist and accumulate further genetic damage, increasing the risk of cancer. While E6 and E7 are the main players, other viral proteins like E5 can also contribute to cancer development by promoting growth signals and immune escape. Additionally, these oncoproteins can influence gene expression through epigenetic changes, further pushing cells toward a cancerous state. Understanding how these oncoproteins work has important implications for prevention and screening. Detecting high-risk HPV DNA and the activity of E6 and E7 proteins forms the basis of current screening strategies. Vaccines targeting these high-risk types are designed to prevent infection and, consequently, reduce the risk of cervical and other reproductive cancers. In summary, high-risk HPV oncoproteins E6 and E7 cause cervical cancer by disabling the cell's natural tumor suppressors, promoting unchecked growth, preventing cell death, and helping infected cells hide from the immune system. This complex process underscores the importance of vaccination, regular screening, and early intervention to protect against HPV-related cancers. Always consult healthcare professionals for personalized advice and screening options to stay informed and proactive about your health.